All right. Good day, everyone. My name is Alan Garner, and today I will be presenting my talk entitled Characterizing Electromagnetic Field Interactions with Biological Cells to Guide Medical Applications. So, the goal of my talk today is to discuss the technology of electromagnetic fields for use for medical applications, particularly for cancer therapy. And while a lot of talks will focus more on the actual applications, the goal of my talk today is to discuss some of the basic physics and biophysics behind these interactions and try to explain why understanding this physics is so important to guiding the practical side of things. So my goal today is to talk about the physics of both pulsed electric fields and a little bit about alternating current fields, or AC fields, to show similarities and differences. So, to start off with, we will talk a little bit about how biological cells behave in the presence of electric fields. So, although you may not typically think of it this way, biology is inherently electrical. For instance, you can take the biological cell here shown on the left, take a standard cell diagram that you would see in any sort of biology textbook, and you see you've got the nucleus, you've got the mitochondria, you've got the outer cell membrane. All right. Now, as an engineer or physicist, we can take this diagram and we can think of it in terms of simple electrical structures that you might study in a first semester or second semester physics course. In other words, we can take that outer cell membrane and consider it as a combination of a resistor and a capacitor. So this resistance that you see here represents the ease with which ions can travel across that semipermeable membrane. So we can consider that 5 to 10 nanometer thick lipid bilayer as a semipermeable structure that allows some ions to enter the cell by passing through it. And we can measure the ease with which those ions traverse the membrane through the resistance. Similarly, if you recall your first or second semester physics course, you may recall the definition of a capacitor. And in its simplest form, a capacitor can be defined as two parallel plates with some material in the middle. That material is referred to as a dielectric, all right, and it has some properties associated with it, most notably the permittivity. So we can take this lipid bilayer and we can represent it as a combination of this capacitance and this resistance. All right. Now, a cell isn't just standing in vacuum. It's usually surrounded by some extracellular fluid. In vivo, this is blood. In vitro, it might be some sort of buffer solution. So the buffer solution contains ions. You can think of these ions as charged particles that can move, and there's going to be a resistance associated with that. So you see the resistance here and here, which represent that resistance. Similarly, if you're thinking about an electron, a cell, and you're going to be applying a voltage to the cell, you're going to be having this suspension containing cells with the liquid and cells in between two plates. So you got two plates with some cell or cells in between, and you've got a capacitance. And you can go on through the cell in a similar way. The cytoplasm is represented by resistance because of the ions. The nuclear envelope is just like the cell membrane, except for the nucleus, and so on and so forth down the line. So when you think about a cell like this, it's important to realize that these parameters are not static. They are dynamic. So if you apply electromagnetic radiation to these biological cells, these parameters change. And they change because the electric fields interact with cellular structures, such as the membrane or the mitochondria or the nucleus, and that winds up manifesting itself from an electrical perspective in changes in these parameters. Before I talk more about that, we can also think of the cell in this way. This is commonly referred to as a two-shell model. There's an outer shell, referred to as the cell membrane. It's defined by its permittivity and conductivity. Permittivity maps to capacitance. Conductivity maps to resistance. Same basic idea. The conductivity here represents the ease with which ions can traverse the membrane. And the cytoplasm, similarly, permittivity, conductivity, nuclear envelope, and nucleoplasm. And then you have these dimensions, which are important in characterizing the biological cell. So when we apply an electric field to the cell, right, we're changing the cellular function. And one of the ways in which you do this is by applying a sufficiently long electric pulse. You can take this biological cell, and as a physicist, right, we've talked about it as a combination of resistors and capacitors. It's fundamentally a membrane charging problem. In other words, you're applying a voltage, and there's a time constant. The time constant is defined by the product of the resistance and the capacitance. And if the pulse duration exceeds that time constant, you fully charge the membrane. By doing so, you create what was initially referred to as dielectric breakdown. So in many of the initial experiments, most notably a seminal work by Sale and Hamilton in the mid-1960s, they applied electric pulses to microorganisms, and they observed a rapid increase in the conductivity of the cells. A rapid increase in conductivity in the electrical engineering perspective is characteristic of what's called dielectric breakdown. You can create dielectric breakdown of, say, plastic materials in your high voltage devices. And what happens is you have a rapid increase in the conductivity of the plastic. Now, in a solid such as that, this dielectric breakdown is irreversible. However, in the case of a cell membrane, that breakdown, quote unquote, may be reversible or irreversible. Now, if you're doing an application like microorganism inactivation, or one of the main topics of this conference being cancer therapy, irreversible electroporation is obviously okay. However, in the 1970s, it was observed that if you could control the pulse duration, right, and the voltage, so you're not completely charging the membrane, but you allow it so that the membrane can recover, you can create what is referred to as reversible electroporation. And some of you may be familiar with transfection, where you try to put GFP or RNA or other molecules into biological cells. That's the concept behind transfection. The technology was then subsequently named electroporation. And the idea is that by applying these electric pulses with sufficiently long duration, sufficiently high intensity, you can charge the membrane. And physically what's going on, based on molecular dynamics simulations, is you're shifting membrane-bound water. That membrane-bound water shifts the lipid bilayers and creates pores. Electrically, pore formation increases the conductivity because you're opening up pores and allowing ions to flow in. And as long as you remove that pulse before you create pores beyond a certain critical threshold, then your pores will reseal and you'll retain viability. So that is the concept behind electroporation. And here's the slide. And a typical way to do this is you apply, this is a simplistic square pulse, an idealized version of the pulse. A typical real-life pulse will have some finite rise time and some finite fall time. Here you see it's very instantaneous. But conceptually, what's going on is you're choosing a pulse duration on the order of or slightly larger than the membrane charging time, and you're applying a sufficiently strong electric field. Typical parameters, a few hundred volts per centimeter to a few kilovolts per centimeter, pulse durations on the order of microseconds to milliseconds, right? And then what you're doing is you're charging this outer membrane. Typical values from the literature, several hundred millivolts to a few volts. Doesn't sound like a lot, maybe, but keep in mind that lipid bilayer is about 5 nanometers thick. So you're creating very strong electric fields across the membrane, even though the voltage across the membrane may be relatively low, right? And here you see some examples. The one here in the upper right is actually looking into what is called electrochemotherapy. So traditional chemotherapy uses drugs to kill cancer cells and tumors, more specifically. However, to succeed in this, you have to use a fairly high concentration of the drug. because the drugs do not easily permeate the tumors. So the concept of electrochemotherapy is to use an array of electrodes to surround the tumor and apply these microsecond pulses in trains and permeabilize the cancer cells to allow drugs to enter the cell. So by using these typically microsecond to millisecond pulses, you can actually permeabilize the tumor, put the drugs in, and even if the pulses are reversible, inducing a reversible electroporation, you'll increase the drug concentration. This allows you to induce the biological effect with a significantly lower quantity of the drug. These drugs are highly cytotoxic. They kill a lot of healthy cells as well. This allows you to minimize the dose but maintain the efficacy. So we've talked about this. I worked for Carl Schoenbach at Old Dominion University in the early 2000s, and he posed an interesting question. What if, instead of this pulse duration you see here that exceeds the charging time, what if we use pulses with pulse duration shorter than the charging time? In other words, we don't necessarily target this outer membrane, but we can fully charge intracellular structures. Why is this important? Well, one of the hallmarks of a cancer cell is that the cancer cell mechanism for programmed cell death is broken, all right? So the cells cannot undergo apoptosis as they normally would. So by targeting these intracellular structures, the goal is that you may be able to permeabilize the mitochondria and facilitate the induction of apoptosis in these cells that can no longer undergo apoptosis. So the objective of the early research at Old Dominion that still continues is how can these nanosecond duration pulses, which are shorter than the charging time of the cell membrane, how can they affect cellular function? So the idea behind this was to take this electroporation pulse, which was microseconds to milliseconds in duration, hundreds of volts per centimeter to a few kilovolts per centimeter, and apply the same energy under the pulse. So instead of applying it over this long period of time, you apply a stronger voltage with a shorter duration, and you're now applying 10 to 300 nanoseconds, 10 to 300 kilovolts per centimeter. So the idea here is that you're not fully charging this outer membrane, although you will certainly still have a membrane level effect, but you're preferentially targeting these intracellular structures. Now, it's important to point out that these pulses themselves are designed to mainly have athermal effects. In other words, you're not intentionally inducing heating of the cells, right? The fundamental mechanism behind this is electrical. It's a membrane charging event of some sort, be it the cell membrane or the intracellular structures. Now, it's important to recognize that whenever you apply an electric field to something, you are inducing some level of Joule heating. So there will be heating associated with these pulses. And molecular dynamics simulations indicate that heating will facilitate electroporation. So the more heating you have, the easier it is to permeabilize the biological cells. All right. The other thing that we'll talk about specifically in this talk is that as you use these shorter duration, faster rise time pulses, and you start to have higher repetition rates, you can also induce, even if you're not creating significant heating of the overall solution, you can induce very strong temperature gradients across the membrane. And we show some of the effects of that through molecular dynamics simulations in this talk. So again, fundamentally an electronic or electrical effect, but temperature and temperature gradients can significantly contribute. So this is a broad overview. This is from a review by Jim Weaver. And what he showed was the level of knowledge and the understanding around 2012. And you have this, what's called super electroporation. He termed that word. When you apply these nanosecond pulses to the cell membrane, remember I mentioned you're still creating pores, but you're not creating the traditional electroporation. These pores, in quotes, are large. Okay? And by large, we mean they're still nanometer size, but they may be several nanometers, like 5 nanometers or so based on computational work, okay? 5 to 10 nanometers. At the very least, they're large enough to allow molecules to traverse. However, nanosecond pulses do not create these large pores because you're applying the pulse and removing it before you fully charge the membrane. So, but they do still create some degree of pores. And Jim Weaver termed this super electroporation because his simulation showed that instead of creating these large pores, you're creating a larger density of smaller pores. So you're not going to be able to get large molecules through these smaller pores. But what you can do is you can actually encourage the transport of, say, ions. And one of the applications that ODU initially worked on was looking at platelet activation. Platelet activation worked through, instead of using bovine thrombin, which comes from cows, they activated the platelets using electric pulses and calcium with the idea that the nanosecond pulses would create these nanopores, allowing calcium to go into the cell. And then you're also using the intracellular effects to release intracellular calcium. Now, in collaboration with GE, we have shown, Purdue and GE together, that you can actually induce the same effects with microsecond. So it's not strictly a nanosecond effect. We're just inducing membrane behavior. So some effects require intracellular effects. Some effects can be satisfied effectively by leveraging longer duration pulses. And that's just one example, even though this is relatively unexplored in the early 2000s to 2012. There are groups that are leveraging this sort of 1 to 10 microsecond regime now. Most notably, Virginia Tech, Rafael Dabalos' group is looking into what's called irreversible electroporation, or H-fire, and their pulse duration now are using this several microsecond pulse duration. And the idea behind these several microsecond duration pulses is you're getting some effects. These millisecond and longer pulses, these are true conventional electroporation. These are the nanosecond super electroporation. But this 1 to maybe tens of microsecond, you're getting some sort of combination. You're getting both membrane level and potential intracellular. So it allows you to optimize your effects, such as for cancer treatment. So what do we use these things for? Well, electrochemotherapy we talked about gene therapy, we mentioned irreversible electroporation. People are looking at nanosecond pulses. Rich Nucitelli works for a small company out of the Bay Area. They're looking at nanosecond pulses for things like liver and prostate. There's also transdermal delivery. So you can see that there's a broad array of electric pulse biomedical applications related in some way to cancer treatment. There's also work that my group has done looking into stem cell stimulation. We've looked into microorganism inactivation with novel combinations of antibiotics. And we've also even looked into bioenergy, looking into extracting lipids from algae and trying to optimize that process. Now, one thing that's becoming increasingly interesting are the effects of different waveforms, not just pulses, but also alternating current and pulsed radio frequency pulses. You may have heard some of the things out in the open literature about what's called the Havana syndrome, where people are suffering these mysterious symptoms that may be attributable to pulsed RF radiation. All of you, I'm sure, have heard of the ongoing discussions about the safety of cell phones. This has been going on for several decades now, looking at these low intensity RF fields and trying to understand what they do to the body. And the jury is still out on cell phone usage and what it does, or if you need a large amount of exposure to cause some sort of effect. It's comes down to a lot of epidemiology. It becomes a very challenging problem. Even more challenging problem that I've seen some discussion on in the literature is Wi-Fi. People are trying to relate Wi-Fi and the ubiquitous exposure of Wi-Fi to depression. Again, these sort of low intensity fields become much harder to gauge because you're looking at mechanisms that become more stochastic and epidemiological in nature rather than strictly a direct biological effect like I just showed with electroporation and nanosecond pulse electric fields. Nevertheless, we can leverage some of what I talked about with pulses and relate it to AC fields. So we're looking here at the general range of AC fields. And I want to draw your attention to a couple of things that we'll talk about. You see here what is colloquially referred to as the RF regime. Some people take the RF regime up to around a couple of gigahertz. It comes down to community and personal preference, but we can refer to RF in a general sense as kilohertz to megahertz for these discussions. Then you can go up to traditional microwave, 2.45 gigahertz, commonly used magnetron from a microwave oven. And there's also increasing interest in biological effects of millimeter waves. A lot of literature from the former Soviet Union, a lot of work in the 60s to the 80s trying to understand millimeter wave biological effects. If you take it a little bit further, there's even a lot of interest in developing imaging in what's called the terahertz regime. Terahertz being 0.1 terahertz or 100 gigahertz up, which is where you see millimeter wave here. A lot of people will also colloquially refer to around the 20 to 30 gigahertz regime as the start of millimeter wave. Okay? So the biological effects at 2.45 gigahertz are predominantly thermal. And I can give you an example, and I say predominantly for a reason. So at 2.545 gigahertz, if you do the calculations, you need a very, very strong applied electric field to induce a membrane potential. Okay? As you go down to these lower frequencies, you can induce a membrane potential comparable to the 1 volts I discussed earlier. Okay? 2.45 gigahertz, you can't do it. Now, the reason I bring up predominantly thermal is that when I was at GE, we found a paper that claimed they could transfect, I believe it was CHO cells, but they could transfect a mammalian cell line with GFP, green fluorescence protein, using a 2.45 gigahertz field with no heating. So one of the early projects that we worked on at GE when we were getting into this field was trying to characterize what could be going on. And this leads to my earlier discussion about temperature gradients. And we worked with a group out of Russia called Kintec to model these temperature gradients. So RF radiation absorption, what's going on? Well, I used to teach RF radiation safety at the U.S. Naval Research Lab. This is one of the slides. Big picture, you can have transmission, you can have absorption, you can have reflection, you can have scattering, okay? The absorption, let's say the RF radiation hits you, it's going to cause heating. And that leads to this concept of SAR, specific absorption rate. And when you're dealing with RF radiation, again, 100 kilohertz to 10 gigahertz, this is what is commonly used for safety standards. So the outstanding question remains, is this the only effect? Do you have non-thermal effects? This is something we're actually looking at now, is we're trying to characterize both thermal and non-thermal effects for various waveforms, including pulsed RF and AC fields. We're at the very nascent stages of the study, but we're trying to characterize that and trying to understand how these effects may play a role together. So SAR depends on conductivity, depends on the electric field, and they have these limits that they came up with many years ago. But again, a lot of the basis for these limits has been lost over time. And this is becoming increasingly important, for instance, from a Department of Defense perspective, as a lot of the ships, I'm Navy in one of my other lives, so as ships start to have more high power electronics on board, you want to understand, is thermal the only effect, and does that allow you to relax some of the safety standards, or do you have to make them stricter under other conditions? So trying to understand those effects. This is what this looks like. Again, you're looking at the electric field strengths you're allowed to have as a function of frequency. Okay, so you can see these sort of effects. This comes from the IEEE standard. Now, as you go down to lower frequencies, as I mentioned before, things start to look a lot similar to what we talk about with pulses. This is some work done by a former ODU colleague, Sunao Katsuki from Japan, where he looked at two different frequencies, or three different frequencies, I should say, 0.02 megahertz, 2 megahertz, and 50 megahertz. And he looked at staining inside the cytoplasm and inside the nucleus. Okay? So what he's trying to show here is that different frequencies, where are the effects concentrated? Okay? And so what he's claiming here is that this increased fluorescence in the cytoplasm and the nucleus at 0.02 megahertz, this is characteristic of electroporation. Okay? Now, at these lower levels, at these higher frequencies, no electropermeabilization significantly is really happening. However, looking at the nucleoplasm fluorescence specifically, okay, what we're seeing here is the effects of these fields on degradation. Okay? So at 1.4 kilovolts per centimeter, 2 kilovolts per centimeter, 1 being the baseline, you're seeing nuclear degradation. So at 50 megahertz, you're getting an intracellular effect. At 0.02 megahertz, you're getting a membrane effect. And in between, around that 2 megahertz I showed, you're getting a little bit of both. Okay? So AC fields at these RF type levels, hundreds of kilohertz, tens of kilohertz, to tens of megahertz, you get membrane charging. You go up to higher frequencies, you're getting heating effects, or as I'll show later, potential temperature gradient. So to understand these phenomena, right, we've talked about this, to understand the physics, we need to be able to characterize these properties. In other words, if we want to use modeling, we need to understand how these permittivities and conductivities behave. And one of the ways that you can do this is through a technique called time domain dielectric spectroscopy. So we've talked a lot about pulses where we're applying these very high intensity fields. Time domain dielectric spectroscopy works by emitting a long duration, 5 microseconds, but very fast rise time pulse, 40 to 50 picoseconds, in very low intensity, about 200 millivolts. And you're shooting this pulse down a coaxial line. Now, the math may be a little complicated, but fundamentally what's happening here is if you have these very fast rise times, when you convert that into the frequency domain, that gives you a very high frequency. So the idea of the fast rise time here is to give you a broad frequency range so that we can characterize the full cell. Because we don't need a very high frequency to characterize the membrane, but we do need a higher frequency on the order of many megahertz to characterize the nucleus. Okay? So what we can do is shoot a voltage down the line, measure the reflection, take out various artifacts such as the conductivity of the suspension and artificial layers that form on the electrodes. That gives us the properties of the cell suspension. And then we can do some math to extract the permittivity and conductivity of the individual constituents. Okay? And this is what I did for my master's thesis at ODU and part of my PhD work at Michigan, was I tried to characterize these properties. And I worked with a visiting professor, George Chen, and we actually measured the change in these parameters over time following a train of 5 10 nanosecond, 150 kilovolt per centimeter electric pulses. What we observed was that the cytoplasm conductivity for the pulse cells went down over time, and the nucleoplasm conductivity similarly went down over time. So what we're seeing here is we're seeing the leakage of ions from inside the cell to outside the cell. And we can quantify it using this technique. What this showed, although I didn't emphasize it at the time because we were still operating under the assumption that this was predominantly intracellular, but other scientists at the time, Tom Vernier, who was at the University of Southern California, and Andrei Pakimov, who was at Air Force Research Lab in San Antonio at the time, they demonstrated that they were forming these nanopores. And that's what you're seeing here. You're seeing the formation of nanopores, and this first measurement was taken a minute after the pulse. You're seeing a pretty dramatic decrease compared to control within a minute because those pores form very quickly, and they have a fairly long lifetime. You see you're starting to get to a steady state around 5 minutes. So those pores have a lifetime of about 5 minutes. And you see the same effects here. You're seeing around 5 to 10 minutes, you're starting to get to a true steady state. Although within error, you're arguably within steady state after 5 minutes. So now I'm going to briefly talk about modeling. So modeling is important to providing guidelines to doing these sort of studies. And the way this is done is by using the Smolakowski equation. I'm not going to bore you with the math behind this. Fundamentally, what the Smolakowski equation does is for an applied electric field, it gives you the calculated number density of pores of different size that are formed within the membrane. So one of the projects I worked on at GE was looking at these RF fields and trying to understand what they did. Another project that I did here at Purdue, before I get into that, is looking at the traveling of calcium. So I remember I was talking about platelet activation. That's predominantly a calcium effect. So what we did was we applied a 600 nanosecond pulse from left to right on your screen. And as you apply the pulse at 300 nanoseconds, what you're seeing here is the concentration of calcium. So you're permeabilizing the membrane, and you're also moving calcium by what's called electrophoresis. So the calcium ions follow the field lines. That's what you're seeing here at 300. At 600 nanosecond, you've brought in a lot of these ions. Now, after 600 nanoseconds, you remove the pulse, and you see the concentration going down. Okay, going down at 1 microsecond. But as time goes on, the pores remain. Because remember, I showed you experimentally, these pores do not immediately reseal. It takes a long time to reseal. So if you do the calculations, you see that the cell has a time constant for diffusion on the order of a millisecond. So that's what you're seeing at 1 millisecond here. You're seeing the ions start to go into the cell. And then at 4.5 milliseconds, you're now seeing a larger concentration. We can also use this for the Smolakowski equation. And what I did here, to try to understand what we may be able to do with the RF fields, we applied an electric pulse to create pores. Then we applied AC fields of different frequency and different intensity. Now, these are on normalized units because that made it easier for me to run the simulation. But what you see here on the left is the average pore radius. These V sub A's are the different membrane potentials. You see that there's a quote-unquote magic membrane potential where the pores neither grow nor shrink. They oscillate about a steady state. As the membrane potential either goes up or goes down, you see that the pores grow or shrink. We can explain that physically by the figure on the right. The figure on the right shows the rate of energy, the pore opening energy or pore creation energy, if you will. On the vertical axis, time on the right. So at a low voltage, you see how that dpdr term is positive. That means that the pores will shrink. If it's negative, as in the case of 0.33, you see how it's beginning to oscillate and become more and more negative. Pores will grow. And then 0.23, you see that it exactly oscillates about the steady state. It's important to note that as a function of membrane potential, this behavior is independent of frequency. As long as the frequency generates that membrane potential, you get this behavior. Now, obviously, as you change the frequency, you're going to have to change the voltage that you apply. As the frequency goes up, you need to apply a higher voltage to get the same membrane potential. Okay? Moreover, I actually derived in this paper in Journal of Applied Physics, I actually derived the condition for this steady state. Using the differential equations that we had, I solved for the steady state and recovered that 0.23 using the RMS voltage. Now, this is great in theory, right? Literally in theory. But in a practical device, if you're applying an AC field, you're going to get heating. So if you were to do this in practice, you'd probably need to do some sort of pulsed RF. Would sound familiar from our previous discussion. And even then, you're just keeping the pores open, but that's okay over time. So you may not get electrophoresis because an AC field, you're alternating the polarity. So you would alternately be pushing and pulling the ions or molecules. However, if you're doing a pulsed case, right, because you're for thermal concerns, you'll keep the pore open, then you'll stop. The pores may reseal a little bit, but depending on your repetition rate, you may get some diffusion. So you may be able to play some games with your pulsing to make this work. that leads to the next thing. So I mentioned before this microwave thing that we're trying to understand. So one of the things that we did was we calculated the temperature gradients. And we assumed this simple Gaussian type of shape shown in A as the temperature. We have boundary conditions and equations. And then here we're assuming a voltage divider. There's a difference in the resistance or the conductivity between the outer suspension and the inner suspension. As long as you have that, you'll get the resulting temperature gradient across the membrane. This shows you the resulting calculation in normalized units. Top is change in temperature as a function of position. 0 is the center of the cell. 1 is the outer part of the membrane. The bottom figure shows the normalized temperature gradient. And we're applying a 10 nanosecond pulse and a 10 microsecond pulse. You see on the left bottom, the 10 nanosecond pulse induces a very high temperature gradient, up to 100. The peak temperature gradient for the 10 microsecond pulse occurs at this 18 microsecond time, and you're getting a gradient of about 2. So you get a dramatically higher temperature gradient using the shorter duration pulse. Okay, so when we talk about electroporation and membrane dynamics, we focus strictly on the electrical effects, but we don't talk about temperature gradients much. And one of the things we hypothesized in this paper is that the temperature gradient could enhance the electric field effects because there is a phenomenon called the thermoelectric effect. Thermoelectric effect says if you get a temperature gradient and you have a polar molecule, a lot of polar molecules in a lipid bilayer, lipids and water is polar. If you have a temperature gradient, you can create a voltage gradient or an electric field. And what drives this electroporation phenomenon? It's that membrane potential or electric field you're forming across the membrane. So we've also looked at the multiple pulse regime. You see an example here. The big takeaway from this slide is that higher repetition rates can make the gradients more uniform, what you're seeing here at the top. But it may not be practical for pulsed electric fields because as you go to a very high repetition rate, you're going to heat up the cells and cause boiling and other issues. But it may be relevant for pulsed RF or other things where you have combined modality. So here is what we look at for this temperature gradient. So working with Jihui Song from Wentworth Institute and Ravi Joshi from Texas Tech, we actually did molecular dynamics simulations where we started off with a subthreshold field. So the left figure you see here is subthreshold, no electroporation. The one on the right has electroporation. You can tell the electroporation by the formation of what's called a water wire. That's those particles that cross over. That's indicating electroporation. So by combining a temperature gradient with the applied electric field, we are now getting electroporation. And we've also started to look at the same sorts of calculations with AC fields. This is one method that I've looked at trying to apply the techniques from the earlier GE work. We didn't get terribly far with this because I was trying to do it on the side and I didn't have a student to work on it at the time. We have a proposal that will hopefully be funded soon where we'll actually continue some of this work. But what we generally saw was that a higher frequency does seem to create a higher temperature gradient. This is an alternative approach that I looked at in graduate school. This was work done by my PhD advisor and some of his graduate students, looking into different types of high power microwave fields and the ability to heat particulates. Okay. And the idea here is that by using AC fields in an appropriate mode, you can create a temperature gradient across those particulates. So we tried to do the same thing. We took a typical pulsed RF field from our research group, okay, and then we did the calculation to determine the change in temperature, okay? And when you put in that change in temperature with a 10 nanometer thick membrane, you get something that looks kind of like this. You get something that looks like around 4.76 times 10 to the 6 kelvins per meter. So again, we are starting to look at these calculations, trying to characterize what exactly they might mean, but But this approach that I'm showing here may be more relevant for an AC field. So in conclusion, what have we talked about? I've talked a lot about the fundamental physics in trying to characterize how electric pulses can interact with matter and with biological matter in particular. Okay? And the idea behind using all these models, right? Granted, none of them are going to be perfect, but the idea behind them is to elucidate the fundamental physics to help guide the practical application. If we can understand how things work, we may be able to tune and optimize the parameters. The applications, some of the ones that we've looked at in my group, cancer treatment, bioenergy, food treatment, non-lethal weapons, or pain remediation. Those are some of the things that we're working on. From a fundamental perspective, I didn't touch on it too much here, but looking at the universality of effects across the electromagnetic spectrum, right? My group has done work in pulsed electric fields. We've done work on AC, not shown here, but we've done temperature gradient work on lasers. We do work in plasmas. What I'm really interested in is synergies, right? Can we combine these different modalities to enhance the effect? And we're starting to get funding to look at this now. Multi-physics. How do we put in temperature-dependent dielectric properties? How do we put in ion flow and heating and temperature gradients? All of these things. How do we account for those to make our models more rigorous and more relevant to the actual cases that we see? And obviously, I couldn't have done this without a large team. You see here all the students who have worked with me on the bioelectric side, a lot of modeling work, a lot of initial experiments, collaborators from Purdue University, my former colleagues at GE, and my colleagues around the country and the world, even from Germany as well. So, with that, I want to thank you for your time and attention and the invitation. And with that, I guess that is the end of my talk. Thank you.